after the completion of the 11th chapter, that is in second PUC biology, let me start the explanation of the chapter number 12, that is biotechnology and its applications. So here the chief objectives of this biotechnology, that is the application of biotechnology, to develop stress tolerant plants for the drought, heat, and then salt, coldness. So that is the one important objective. And then disease resistant plants and pest resistant plants, and enrichment of the vitamins like A, C, etc., and the increase of the efficiency of the minerals, proteins, oils, vitamins, minerals in plants. So these are all the various objectives of the biotechnology applications. And coming to the, as we know very well, the golden rice, in which vitamin A is enriched. The earlier the rice cultivated, lack of vitamin A, deficiency of vitamin A, that was enriched by producing the gold rice, that is golden rice, which is yellow in color and rich in vitamin A, and that has cured the blindness in several children. So these are all some of the examples. And then coming to the pest resistant plants, to develop the pest resistant, not only in BT cotton but also in tomato, potato, and then soybean, etc. So the disease resistant and pest resistant plants, especially the BT cotton. So these pest resistant plants, they develop resistance to lepidopterans members like the tobacco budworm, armyworm, and even coleopter beetles, and the dipteran flies and mosquitoes. They develop resistance to all these the pests or the insects. They contain such plants contain insecticidal proteins, the proteins which could kill the insects. So coming to the BT cotton, BT expansion is Bacillus thuringiensis cotton. So this BT cotton, from this from Bacillus thuringiensis, the toxin protein coded by the genes, toxic coded genes extracted from Bacillus thuringiensis, that is called Cry1AC, Cry2AB, and Cry1AB, three protein encoded genes. They are the genes, protein encoded genes, which are toxic. Three genes can be obtained from the Bacillus thuringiensis and bacteria, and then injected into the cotton plant. So it doesn't affect the cotton plant because of the vary in the pH. When some insects feed on such cotton, BT cotton, these toxic substances enter into the gut portion, and in the alkaline, because of alkaline pH, they become toxic. They produce toxic substances, and these toxic substances bind to the epithelial tissues and make perforations in the gut portion of the insects. Such insects will not survive. Eventually they die. Especially the Cry1AC and Cry2AB, they prevent the bollworms. They are used against the bollworms. And Cry1AB against corn borers. So here the vector for this used the plasmid of Agrobacterium tumefaciens. Plasmid of Agrobacterium tumefaciens. So this is about the BT cotton. And coming to the another one called, so the pest resistant plant that is developed in Nicotiana tobacco, member of Solanaceae member. That is the tobacco plant. There is one parasite nematode called Merodigine incognita. Merodigine incognita. So this usually attacks the roots of the tobacco plant and destroys the roots, and eventually the whole plant dies. So this nematode, to resist this nematode, a novel strategy applied by the scientists, that strategy includes RNA interference. They developed a double-stranded RNA which silences the mRNA so that the mRNA could not produce enzymes required for the digestion of the Merodigine incognita. Merodigine incognita. The source of this can be obtained from the RNA genome, and a mobile which has genes which shows mobility, that is called transposons, can be obtained from this. And the vector used for this one is also plasmid of Agrobacterium tumefaciens. The vectors are very important as far as this application is concerned, and that is the plasmid of Agrobacterium tumefaciens. And these the vectors, they carry the genes which could produce the double-stranded RNA which silences mRNA so that enzymes are not produced, and such the Merodigine incognita will not survive in the plants. So coming to in India, about 30 recombinant therapeutics developed by the India, but the 12 only used in the markets. Next to this is the insulin. Insulin is the hormone secreted by the beta cells of islet of Langerhans. We know very well this insulin. It regulates the blood sugar level in human beings. So lack of this insulin, it causes diabetes mellitus. So the genes first obtained from the pigs and other cattle caused allergic disorders. Hence, the genes has to be obtained from the caused allergic disorders, and there are two genes which are responsible for the production of insulin, that is A gene and B gene. A gene and B gene. The first produced insulin contained extra stretch, and it is called C-peptide. And in 1983, a company called Eli Lilly Company of America developed this insulin with the two genes producing the two chains of proteins, that is A gene and B gene. And the vector here used for plasmid of Escherichia coli, and these A-B chains joined by disulfide bridges. That is about the insulin. And coming to in 1990, gene therapy, that is the replacing the fault genes with the correct genes is called the gene therapy. So first gene therapy introduced in 1990, that is used against a disease called severe combined immunodeficiency disease, wherein the patients lose their immunity. Severe combined immunodeficiency is caused because of the they fail to produce ADA, adenosine deaminase enzyme. This, when the ADA is not produced, they suffer with the immunodeficiency. But in 1990, gene therapy replacing the fault gene with the correct gene introduced, and then that has been administered over a 4-year girl. because by bone marrow transplantation, and the vector used retroviruses. That is also very important. Vector used retroviruses, and when administered and joined to the lymphocytes, it's only a temporary cure, as the lymphocytes are not immortal. So the next thing, as they are temporary, and then hence periodic infusions required to correct this. It is really a difficult problem. Hence, it has to be administered at the embryo level, earlier, embryonic, for the permanent cure. And then how to diagnose a particular disease by two techniques: PCR technique, polymerization chain reaction technique, and ELISA technique, enzyme linked immunosorbent assay. Sorbent means abnormality and assay means estimation. Enzyme linked. So this we can detect it by the presence of the antigens, that is the proteins and the glycoproteins, and also the presence of antibodies. With this we can diagnose, detect the diseases and all. And such animals in which genetically modified recombinant DNA used, called GMO, genetically modified organisms, are the transgenic plants and transgenic animals. For which there are different steps used. Usually normal physiology development will be observed in such plants or the animals, and the study of the various diseases. Say for example, the rheumatoid arthritis, Alzheimer's disease, cancer, cystic fibrosis, and all emphysema are the various diseases which has to be studied by the scientists. And the biological products produced by these animals. For example, alpha antitrypsin produced to cure the emphysema, and attempts have been made to cure the diseases like phenylketonuria and cystic fibrosis. That is in 1997, attempts made to cure. This is also very important. The years also very important in this aspect. And the testing the vaccine for its safety, and then the again clinical safety testing and all these things. These are all the steps included in the producing the transgenic animals and the transgenic plants. And finally, there will be one committee looking to whether it is right or wrong. That is GEAC, Genetic Engineering Approval Committee, GEAC established to see whether how far the vaccines are, the safety, etc. And then in India, about 2 lakhs varieties of rice will be cultivated, produced, and also 27 varieties of basmati rice. But in 1997, America patented the basmati rice. India patented the neem and turmeric. And using of the resources of one country without obtaining permission, called biopiracy. In case of biopiracy, if there is any biopiracy, means case can be filed in PIL, that is the public interest litigation case can be filed. So recently the Parliament of India has been amended. Amendments have been made by the Parliament, that is emergency provisions. Say for example, in case of corona and all those things, if they patented, if one country patented, not given to another country, the emergency provisions should be made for the research and development, etc. And then the patent terms also should be revised. These are all the things about the chapter 12, which is very, very important, that is biotechnology and its applications. So thank you. Please subscribe and don't forget to subscribe. So with this I have completed about 12 chapters.